Today I'll tell a little bit of the history of ChEMBL and then a couple of stories where I guess cheminformatics and bioinformatics has provided insight into some of the problems I'm sort of interested in. But fundamentally I'm a data guy. I love digging out data and I love analyzing data. And it's great to see the impact that some of the tools that we put together have had. So I'll outline a little bit about my current employer, the Medicines Discovery Catapult, talk about ChEMBL, SureChEMBL and UniChem, a little bit about this sort of errors, sort of panic that you get really everywhere in science. Every aspect of life science is going through a sort of bit of a confidence crisis in confidence. And probably if you're developing methods that require perfect data, you're up to no good because you'll never have that in the real world, I think. So part of it's just curating data, finding the odd stuff and so forth, and dealing with real life. One of the other areas I'll talk about is competitive intelligence, where actually I personally use quite a few of the ChemAxon tools to do some of this work, and this is sort of trying to, to put together a good view of the drug discovery landscape, in particular the sort of relationship between target and chemotype. Then our antibacterial is really different, and then a tiny bit of recent work around assay networks, the concept of building a sort of predictive network of assays that should be tried between every single target and every single known human disease. And I had to leave some stuff out. There's some slides at the end. You're happy to take the slides, the final version of the slides away, about drug blending and some recent work I've been doing around, I guess, tackling resistance from a sort of cheminformatics viewpoint. So the Medicines Discovery Catapult. So the UK is interested in making a series of investments to keep UK industrial capacity and capability running at full speed. And so Innovate UK, a government agency, have funded and spun out a series of these so-called catapults. I work for the Medicines Discovery Catapult. There's also a very successful cell and gene therapy catapult, working primarily on manufacturing and producing next generation cell-based therapies. There's a precision medicine catapult. So these are the sort of life science ones, but there's also a whole bunch of really cool things like a satellite catapult. My 8-year-old thought this was a big rubber band that you pulled and fired things into space. But it's really sort of trying to capitalize on the UK's expertise in building and deployment of satellite and space-related technology. So the Medicines Discovery Catapult's been running for about 6 months. I've been there for 3-4 weeks. So it's a new organization, and we're just starting to plan about the type of things that we do going forward. The catapult infrastructure in whole's almost got €1 billion of operating budget, 600-700 academic collaborations produced, about 2,500 industry collaborations, and support of 3,000 SMEs. And really this sort of angle or axis here is what the Catapult program is about. It's taking ideas from academia and trying to get them into SMEs and trying to give SMEs as much support as we can to get them off the ground, up and running and working independently. So there, unfortunately, there's a large number of large, unused or relatively underused pharmaceutical buildings in the UK. So we're based in the old AstraZeneca site. This is our building here. So this is at Alderley Park. One of the big reasons for joining was the lake at the back, which I can sneak off to at lunchtime and do some angling. But what we're building is a national centre of applied R&D expertise to promote faster patient drug discovery and really helping from preclinical all the way through to clinical medicines discovery. Working with companies of all sizes, and academic researchers, but really with a big focus on SMEs. So we'll develop technology, we'll develop some informatics approaches, we'll do some virtual discovery, and partner with charities to try and think of new ways of financing drug discovery. So Medicines Discovery Catapult is a UK not-for-profit company, so any profits we make will be ploughed back into the business. We plan to spend quite a bit of time developing disease-based syndicates, so bringing together stakeholders around patients and particular disease areas and trying to start up where we can find some space for us to operate new drug discovery programs. Getting things into clinical trials is really as important as possible. And I think this is one of the other things that informatics approaches can really start to build, again, from a data perspective. I think the clinical aspects of data available for drugs and for target validation and so forth is pretty poor at the moment. But if you're trying to discover drugs, the properties of those compounds is really crucial to understanding whether you do actually have a business and something to work with. So we'll do wet science. We've got some labs. We've got a lab with a solid-state NMR facility at the University of Warwick. We'll build some informatics. We'll do some virtual discovery and also some technology development. And really, again, another way of phrasing what we're trying to do is lower the barrier to entry. So if people have got some big, expensive experiments to do, we'll try and get them or let them do those experiments as simply as possible. If they want to do some informatics analysis without having to recruit a large team and sort of inbuilt setup costs, we'll try and help. And you're looking at the informatics department at the moment. But in about June 2018, we'll have about 25 staff. So we'll have enough developers, analysts, and data scientists to do some reasonable sort of heavy lifting in this space. So ChEMBL, SureChEMBL, and UniChem. So probably most people are familiar with ChEMBL. Does anyone use UniChem? Oh, brilliant, 3, 4 people. That's the biggest I've ever seen. So that's good. So UniChem I think is underused. But the reality is there's probably quite a few other sort of chemical resolvers out there. So ChEMBL, it is the world's largest primary public database of medicinal chemistry data. Originally started off as a sort of technology asset of a company called Informatica that I was an early employee of. We built a database of medchem, at that time containing about 200,000 compounds, and it was a pretty good asset. The company, as in all of these things, comes to a bit of a sort of fork in the road, needs to make some strategic choices. And the Wellcome Trust sort of spun us out from Informatica. We were actually held by Biofocus at that time, and set up Starlight at the EBI. We called it ChEMBL because EMBL were the sort of employers or the organization that we worked for, so we wanted some sort of brand name. And ChEMBL's grown from that 200,000 odd compounds to now 1.7 million, covering about 11,000 targets and about 14 million bioactivities. It is truly open data, so it uses the same license as Wikipedia, which is arguably the gold standard in keeping data out in the public domain and survivable. And I think it's been a great success. I'm sure it wasn't put up just for my benefit there. Some of the ChEMBL screens outside. People are using ChEMBL all the time. I think it's got a really good sort of place in the world. Because it's open data, it's loaded into other resources. Yeah, the number of papers now, though, that I get to review that are essentially just ChEMBL reskinned or re-indexed in some other way and so forth. So yeah, maybe that's just part of the way the ecosystem grows. People try and fork stuff, add value and so forth. Some of those things will survive. I think the ChEMBL virtual machine, my ChEMBL, is a great little bit of technology. In principle, you can get it on an 8 gig USB drive. One of the crazy programmer guys in the lab had it running on a Raspberry Pi. So full chemical structure searching, 1.5 million chemical structures at that time. On a Raspberry Pi, I think is quite a fantastic thing to be able to do. And many years ago, 10, 15 years ago, data of the scale was only really available in large pharma companies. Putting it together, putting it out in the public domain, I think was an important thing to do. And of course, there's other sort of formats you can download the data in. The data comes from manual abstraction of the primary literature. At the time we started to do this, there was an alternative to manual extraction. So the chemical structures often aren't represented in a nice, easy-to-find form. They often require conversion from diagrams or from names. The assays don't have convenient little uniprot tags and so forth in the assay descriptions and so forth where they're reported. So a lot of work, a lot of expertise needs to go on in doing that. However, I think we're entering an incredibly exciting time for name entity recognition and natural language processing in general. And my vision is in 5 years' time, resources like ChEMBL will be able to be built and curated by little robots, little agents working away and flagging a relatively small subset of data for clarification by experts. But fundamentally what ChEMBL does is stores bioactivity data, the endpoints against either target-based assays or phenotype type assays. And a nice advantage, we'll come into this later, is that you have a sort of relationship between the target and the functional outcome of that. And the assays are classified into things like ADMET, functional, cell-based, ex vivo, and so forth. And again, we'll revisit that. You can do some pretty neat analysis. So this is the efficacy targets for drugs, for current approved drugs, small molecule drugs. And I think one of the surprising things for me, at least in this, is that drugs aren't nearly as potent as we think they need to be. Like 100 nanomolar is pretty respectable for a real drug. And also, it's currently in a set of crappy little spreadsheets at the moment, but there's some data around selectivity. And selectivity, we often obsess, well, we need like an ERK2 selective compound and so forth. Selectivity on real-world data, real-launched drugs, isn't often as big as we think it needs to be. Often 15, 20-fold is enough selectivity to get usable differentiation between competing pharmacology. There are some incredibly potent drugs. There are some incredibly weak drugs. One of these days I'll fix this typo. But they tend to be goofy structures, so that things at this end are things like hydroxyurea, really small molecules. And here there's like goofy sort of steroid vitamin D-like structures. But typical drugs, the sort of things you would design, often they're not as potent as we think they need to be. So SureChEMBL, again, I'm just stunned at the impact that this data has had on the field. Really, this sort of unlocking and giving people data to work on, I think, is really transforming things. Number of sort of technology partners here. Probably foremost is some of the ChemAxon tools in the background producing this regular, pretty damn reliable feed of automatically extracted chemical structures from the patent literature. 15 million chemical structures updated daily. There's a full chemistry download and a little, on the download site, there's a package there where you can effectively just replicate the fully indexed database from the data. And I think this is a sort of bridgehead between the sort of resources that were built by manual extraction, careful experts, to the sort of future where computers have a larger role in indexing and extraction of information from documents. So UniChem is a very simple INCHI-based integration service. One of the things we were reasonably regularly asked by partners and so forth was, was this structure really known in the public domain on the 8th of October 2006? Relating back to patent claims and so forth. So one of the things we wanted to do, it's not really sort of obvious with UniChem, is provide a sort of chemical time machine to sort of have a snapshot of the chemistry world at some particular point, to track the addition and deletion of chemical structures over time from various resources, because it's pretty damn difficult to go back and find out when PubChem loaded a structures, not criticizing PubChem, but not all of their dates actually correspond to the real dates when something hit the road. So 140, well, almost 150 million structures from about 30 sources. The very, very simple standard INCHI-based lookups and a series of sort of extensions on that. Covers available chemicals and has some of the sort of functionality of this chemical structure sort of time machine sort of capability. And it's pretty neat. So some personal perspectives on ChEMBL. So I was ChEMBL, well, a little bit of my career history, 9 years at Pfizer, roughly 9 years at Informatica Biofocus, building technology, and then 6.5 years at the EBI as principal investigator of ChEMBL, where we also brought this SureChEMBL project on. So one thing I didn't stress actually was the sort of benevolence of digital science in donating the fully working and pretty functional SureChem product, and that was a sort of pleasure to sort of integrate into our infrastructure. Things that worked well. So a single major visionary funder, the Wellcome Trust. If we'd have needed to get a consortium together, it wouldn't have been the same. I think one of the other strategic decisions that we took very early on was to focus on the data content in the back end and getting the data out there, as opposed to locking stuff behind a sort of proprietary GUI that just drives traffic to our sites. There's some really cool sites out there, but I hate this myself. I hate this sort of GUI candy. I want to get the data and just play with it, and so forth. So for me, that's a key principle. I think balancing the effort between the bioinformatics and the cheminformatics communities and work as well, or fields, was sort of pretty important. A very clear, unambiguous license, same as Wikipedia, as I've said. You look at the licenses for a lot of other resources are ambiguous. It's not clear what you can do, and so forth. It's pretty clear. The other thing is privacy and security from the get-go. So everything, all the ChEMBL services, I think this is true. If it isn't, I'll find out and feel ashamed. But everything we've ever run in a production ChEMBL setting has been HTTPS, so encrypted, client to server, and so forth, which is again an important sort of feature. I think we're pretty opportunistic. The sort of donation of SureChEMBL is a sort of example of that. I think the open data in ChEMBL has reinvigorated cheminformatics research. There's a whole bunch of things that you just couldn't have done at scale in the past without the ChEMBL and the SureChEMBL data. Things that didn't work so well, when you sort of put together the grant, crowdsourcing, we're going to get everything all indexed up and curated by experts. In our hands, that was a bit of a sort of disaster. And whenever I see crowdsourcing and community interaction and stuff, I tend to think it's not going to work. That's my gut reaction. Very few examples of that. Publisher interactions, again, it would be great to get access to more material and so forth. We had to pay for access to material. It sounds outrageous, I know, paying for something. But it would have been nice to have got donations. Royal Society of Chemistry were an exception to that rule. They donated subscriptions to some journals for us to sort of index and put into ChEMBL. Of course, they end up with links back to their journal articles, which hopefully will drive some business. So it's a sort of virtuous circle. I think we had, in my personal view, insufficient staff in outreach and training. There's a lot of opportunities, and following those all up is pretty tiring if just one or two people do that. And again, I think we should have probably prototyped the MyChEMBL, which is fully free, open source software, a lot earlier, and that would probably have driven a bunch of different sort of technology choices for us going forward. So errors everywhere. I used to love it when ChEMBL first came out, and I'd go to a meeting and people would show me loads of errors in ChEMBL in a presentation in a session I was talking at. I used to remind them, we got a way of feeding back to the database this. But over time, I think ChEMBL's got better quality. We've certainly listened to a lot of the feedback of users and try to fix errors. But the trouble is, one of the big problems is that the errors are just sort of baked into the system. So these are two really important papers. So there was a lovely comment article in Nature where an industrial group showed that a whole bunch of preclinical reported data didn't replicate in an independent setting. And then there was a follow-on article in Nature Reviews Drug Discovery where, again, that they showed how much can rely on all this preclinical supported data and so forth. And this was a bit of a sort of wake-up call to the field. People always said, well, that work doesn't validate or whatever. And there's now also a similar crisis in clinical data. So trying to pull together high quality, reliable clinical data is again a really big challenge. So there's a couple of things to sort of realize here, of course, is that science used to work. So scientific insight was built on people reading the literature, acting on it and developing new stuff. So it's not like the error's so big that it stops us making any progress at all because science works. There was a lovely little nature here. And then with the best piece of irony ever in the history of the universe, in my perspective, there was an editorial note underneath this letter saying, hey guys, don't freak out, to say that actually this paper, there was an editorial note to say they had concerns over the reproducibility of the argument of the paper introducing this reproducibility because the data wasn't available for this paper. So like recursive sort of doubts on the reliability of both, well, of papers reporting the reliability of the unreliability of data. So a couple of people, well, many people have looked at errors in ChEMBL. Things follow a sort of a fairly predictable pattern. The errors in compound structures, which are relatively complex, are quite high, so around about sort of 4 or 5%. Target assignment, 2-3%. Activity values, so like 1.6 nanomolar, around about 1%. And activity type, IC50, Ki, and so forth, almost negligible. So the simpler the endpoint, the lower the error. Lots of errors, well, relatively large amount of errors in the in chemical structures, and they're pretty constant across these databases. Arguably, ChEMBL beats these other databases in apart from here, and there we'd argue that we actually had a more sophisticated target model where we allowed the inclusion of complexes, so many drug targets are actually, say, CDK2 with cyclin D in there and so forth, and that was confusing with respect to the way they measured some of their accuracy. And also there's often quite a bit of variability for early data around exactly what subtype of muscarinic receptor was screened and so forth, and we had a way of treating that. In hindsight, I probably would have had a different way of presenting that in the database, but I think fundamentally we took the right early decisions. SureChEMBL, there was a nice paper published as part of the OpenFacts project where the reliability of structural data extracted from patents was measured across a large number of patents against the gold standard, which is the sort of CAS SciFinder system, and ChEMBL, SureChEMBL stacks up pretty well. I think again there, the likelihood of missing a sort of framework structure or finding a similar hit on the basis of multiple extracted structures from a patent are pretty slim. So I'm a fan of SureChEMBL. I use it in my sort of day job now at the MDC, and yeah, it's a really useful resource. So going back to the sort of errors or differences, so a PhD student and myself published a paper a few years ago now where we were initially exploring intraspecies assay variability. The problem we had was when you give a compound to a mouse, why do you often get different results in a human or primate and so forth? So is this down to differences in the pharmacology at the target level, a sort of molecular recognition sort of component between the mouse receptor or enzyme and the human receptor or enzyme, rat and so forth? So this is a scatter plot of the same compound, same target nominally between rat and human sort of orthologs. So this is the binding constant against humans, transform binding constant against rats. There's a pretty good correlation, so science sort of works. Potent compounds against human targets tend to be potent against rodent targets. And then this is the sort of distribution, the sort of off-diagonal sort of spread of these data points. This is a little bit smoothed. One of the interesting things is there's these, in the raw data, there's these satellite peaks at -3 and +3, and this is because of essentially errors in publication typically. So people mislabel the micromolar, nanomolar levels, and this is even done when people deposit data into sort of online sort of databases. So people just get a little bit confused with micro, nano, milli, and so forth. Also, the other type of place you often find this error is where people do a sort of a log transform, setting micromolar as a sort of zero offset, and so the sign, if it's less or weak potent, log potent to a sort of zero micromolar level is just a recipe for disaster. One thing I meant to say about this is that we went back and looked at a number of cases at errors in chemical structures. So this 4% error, could we make it a little bit better? And if you look at the original publications, that's the sort of typical error rate in the scientific literature. So people mess up or misreport, especially in the early literature where structures would have gone through multiple different hands and typesetting and so forth. That's about the error rate in the literature. We fix some stuff, but we also sort of mess some stuff up as well as part of the sort of manual curation process. So after looking at the rodent data, we thought, well, it's a nice control experiment. Why don't we just look the same compound, same target, same species, but from different labs? This is the data from ChEMBL at the time. So this is now comparing either rat-rat or human-human data from different papers. And it's a pretty similar sort of shaped curve. So intra-lab variability or inter-lab variability is roughly the same size as interspecies variability. There are some nice examples of cases where there are genuine pharmacological differences between human and rodent sort of receptors or enzymes. They're pretty easy to spot with this sort of filtering. But, to my mind, when you're doing target generation models, you should pull, at the very least, the mammalian data into a single set for training a predictive model for the human protein, unless you've got good evidence otherwise. ChEMBL also stores a lot of other data, so cell line screening data, so a lot of the data that people work on from ChEMBL, arguably, and process and develop methods on is the sort of biochemical, it's the target compound stuff. But there's also a lot of cell line and in vivo data in ChEMBL as well. This is sort of fairly typical of two very large, very well-funded sort of experiments effectively doing the same thing, taking a bunch of cancer cells, profiling a set of known standards against them and trying to come up to some insights into genomic sensitivity of protein kinase inhibition or whatever. So two big high-impact articles. Another high-impact analysis a little bit later that showed there was effectively no correlation between these two data sets doing nominally the same experiment. So there was subsequently a bit of a debate as to where the differences came from and so forth, but you need to be very careful of using and relying and trusting on this on single large data sets. And I think this is one of the advantages of bringing the data together, in that you can see the same endpoint and compare it across different sources. The other thing which I think is really interesting to look at, it's pretty difficult to come across, is the sort of primary data. So we all assume that the stuff published in JMed Chem and so forth is pretty tight, and even it's going to be N of 3, but the variance on that N of 3 was pretty small and so forth. Real world data is pretty noisy. This is some data that a large pharma company was forced to disclose as part of a patent dispute, and it shows that real primary data for secondary endpoints, dose response, again is pretty noisy. The other thing which I think arguably is getting worse is the sort of incorrect chemical structures around standards. So there was a nice example where crystallography found that the wrong sort of positional isomer of bosutinib was being used in a bunch of experiments for profiling and so forth, and it was really only determined once some structural biologists saw a bit of anomalous density for a chlorine, I think it was, in the wrong position of a ring. The other more insidious error is in cases like this. So again, this is a PI3 kinase inhibitor. A number of vendors, I guess, guessed what the structure of this would be, started to sell it. It started to be accumulated in the literature, so people were buying stuff, labeling it as voxelicib or XL765, contaminating the literature, whereas the real structure was something very different. For the gold standard kinase set that I'll show you in a second, the error rate of that is about 2%. So 2% of the compounds you get from vendors have almost certainly the incorrect structure compared to the real thing developed by the original company. And of course, this is effectively impossible to catch once it's out in the literature. Which leads me on to this sort of competitive intelligence sort of field. So I guess a lot of drug discovery is around clear privileged families. You know, we know that drugs work through a relatively small number of protein families, GPCRs, kinases, nuclear receptors, and so forth. And these target classes do seem to be privileged with respect to the amount of effort you put in on the chemistry side. You can see here, just for example, so GPCRs, 18% of the content in ChEMBL, yet 30% of the drugs. There's a lot of kinase data in ChEMBL. Yeah, there's a little bit of a depletion here. Proteases, or at the very least, endopeptidases, are a bit of a graveyard for drug discovery. It's easy to get inhibitors against them, but very difficult to turn them into drugs, it seems. So there's a lot of interest in understanding and leveraging these particular families, so protein kinases, GPCRs, and so forth. Because there is something in this concept of druggability. There's some privileged binding site architecture and properties that are associated with a relative ease. Drug discovery is never easy, but a relative ease of finding ligands against them. So one of the things I've been working on is sort of rebuilding from scratch a pipeline to assemble in a class-based way compounds in the clinic that are active against a particular protein family. So this is some data for some protein kinases. So I've built a pipeline to search, extract, and validate gathering data. The moment in my database, I've got 683 clinical stage human protein kinase inhibitors. So these are things like gefitinib, imatinib, and so forth. And this is way in excess of what I can work out is in some of the commercial systems. A really interesting thing is over 10% of these now come from Chinese originator companies. And getting the data for those is pretty tricky. And was a sort of bit of a sort of 3-month diversion in working things out. So I think we've got to think that quite a lot of data and disclosures and information won't come from the classical data sources. There's now 41 worldwide approved small molecule kinase inhibitors. Only 34 of these are approved in the States, and a sort of breakdown of the various highest development phase and so forth. So it's a large set of structures, almost 700, that you can do some pretty neat sort of cheminformatics on. The other thing which again, as you put the data together, this is just using a simple call to Unichem. 153 of these, actually we know in complex with their target structures, which again, goes to show, I think, the power of structural biology and the importance of structure-based methods in either rationalization of selectivity and so forth, or importance in design. This is an overnight graph of the database coverage. So the fraction of content in this sort of gold standard set, CAS registry, about 3/4, but not everything in CAS has a structure. And I don't know the structure of all of these, so I know the structure for about 70%, 30% are still non-disclosed. CAS falls down a little bit, but it's a pretty good resource, as you'd expect. SureChEMBL does a good job of capturing these, because the vast majority you'd expect to be disclosed in the patent literature at some time. The difficult thing is sort of from the 10 bazillion structures, he says, with a remarkably accurate scientific estimate of the content of SureChEMBL, finding the one that is this particular research compound from a company. ChEMBL does a pretty good job. Roughly half of the data you've got bioactivity data points in ChEMBL for. Uni is a sort of FDA registration by the Uni SRS is a sort of regulator registration system. Not everything is in there, of course. An interesting comment is for only about 80% are there registries in clinicaltrials.gov for these agents in development, despite the fact that there's clear rest of world content there. So clinicaltrials.gov is great from 2006 onwards with about 80% coverage. Prior to that, it's effectively useless if you're trying to find, it's a little bit unfair. It's effectively useless in trying to find data on early compounds. Guide to pharmacology, about a third. As I said, protein structure coverage is pretty good. Drug bank, clearly a big focus on launched agents. It's relatively modest, and a lot of these. So Guide to Pharmacology, Drug Bank don't have all approved protein kinase drugs in them. I think ChEMBL does. You can do some pretty neat sort of cheminformatics. I'm sure you're going to see a lot of swanky algorithms over the next couple of days. This is some of the work that myself and a student have done recently. So, property plots, spider plots on particular properties of sets of compounds and so forth, thinking about, kinase inhibitors that access particular portions of the body, scaffold analysis, patent coverage and so forth, scaffolds and things. But you can, once you've got the data together, you can do a bunch of potentially interesting stuff. This graph shows the number of approved protein kinase drugs. So it's still a very active area of research. This is the number of non-proprietary names applied for. So this is effectively companies serious enough about developing and marketing a product that it gets a non-proprietary name. And this is a harbinger, so it comes before the approval of a drug. But you can still see this is in a very active growth phase. And with the SureChEMBL data, you can get a view of the sort of patent dates of those compounds as well. I don't have the time or the intelligence really to explain some of the offsets between approval, publication, non-proprietary name, and patent date, but there's some really interesting trends in there that, to my look at the data shows that either the path from patent to the clinic is genuinely getting faster by about 18 months over a decade, or people are strategically patenting later in this field. And I think, getting between those two hypotheses, discriminating between them will be, and of course both could be true, could be an interesting area of research. Productivity as well. One of the great advantages of putting together this sort of data is, in fact, that actually sort of Zipf's law is followed. So a small number of companies have the majority. So 11 companies, I think, cover 50% of those development candidates, and then you can do some nice little plots to show that Pfizer, Novartis, GSK sort of on trend. So this is number approved versus number that they put in the clinic. AstraZeneca lagging behind a little bit, and poor old Lilly here, 19 shots on goal, and to date not a single approved protein kinase inhibitor and things. And again, the more you get this data out there, the more you can sort of debate it and stuff. It's quite interesting in my view. So, our antibacterial is really different. So one of the dogma in the field, I started to get interested in antimicrobial resistance and properties of antibacterial drugs. It's now a major focus for our work at the Medicines Discovery Catapult. It's an area where there's relatively little engagement from large pharma, yet a pressing current medical need, and arguably an incredibly important future strategic need for society. If antibacterial stop working, we really are in trouble. I would have died more than a cat, actually. I've probably had life-saving antibiotics 9 times in my life and things, and I guess the majority of you wouldn't be here now without them, which is quite a scary thing. So part of the dogma in the field is that antibacterials are different to the Lipinski rule of 5. They're often orally absorbed and so forth, but they're in a different region of chemical space. And it's often posed as one of the reasons why, when we did a genomics-led approach to drug discovery from antibacterials, this sort of finding pathways that are unique to bacteria, picking a target, screening it against compound collections. And a large number of companies now have published to show that this was a non-effective strategy. Nothing really came from this sort of approach. So antibacterials are largely larger, more polar. They're mostly natural product derived and seen as exceptions to the rule of 5. And quite rationally, it led to efforts to try and enrich compound collections in stuff outside of the Lipinski space. So larger, more polar compounds on the expectation that they would be pretty good as antibacterials. So I was doing some curation one day at home and assigning targets to antibacterials. And a sort of pattern occurred to me. So this is penicillin. It binds a protein, a family of proteins called penicillin-binding proteins that are sort of bacterial derived. This is the way it works, or believed to be the way it works. It's a natural product, an amino acid derived natural product. And this is another drug, tetracycline, oral, and again natural product derived. But this works through binding a ribosomal subunit. And as I was looking at the data, it just occurred to me that the small drug-like stuff tend to bind to the antibacterial proteins, whereas the larger, more polar stuff tended to bind to these ribosome species. And I thought, well, maybe actually there's a different trend in the data that's been overlooked in the past. Can we use the sort of class of data that's in ChEMBL to sort of get an insight into this? So we published a small part of the analysis a couple of years ago now, where we discriminated between compounds that bind bacterial proteins and compounds that bind bacterial ribosome species, and this explains by far the largest amount of variance in the difference in the chemical space properties of antibacterials. So it turns out that human protein-directed drugs are pretty similar in the properties to bacterial protein-directed drugs on average. Maybe they're a little bit more potent, but that could reflect the fact that they've just come from natural products, as opposed to being anything baked into the structure of the targets. But the bottom line is the big, goofy, polar stuff tends to bind to the ribosome. And when you look at the structures, it's pretty obvious why this trend may well be true. If you look at the composition of proteins in terms of say the element count in the number of element count in the sort of composition, you can see far less lipophilicity in RNA targets in general, far higher polarity, so you've got the phosphates and the charged oxygens, of course, but a lot more nitrogen hydrogen bond donors and acceptors. So the RNA targets are going to be a lot more polar than protein targets. And I started to do some binding site comparisons, and they've just ended up tied away on the laptop. But I'm pretty convinced that when we look at the actual binding sites of these, it's not like the binding sites of RNA targets tend to be as lipophilic and carbon-rich as the binding sites on normal protein targets. So I'm pretty convinced that the fundamental reason for this difference in the polarity and size of molecules is due to the fact that they target ribosomes or RNA targets as opposed to protein targets. There's a nice little paper from Merck, published last couple of years ago now, that sort of supported this view. It's not as if every RNA target will be a ribosome in bacteria. They found a nice little mechanism with one of these sort of riboswitches, druggable non-coding RNA. That's a bit of a sort of clickbait headline from Nature on the cover there. But I think targets aren't always going to be protein derived. Maybe not even for humans. So some of the implications of this work is that there's a clear difference in the phys-chem of antibacterials. In my view, the basis of that difference is largely down to the sort of target class, and some initial structural analysis supports this. What this really means is if we enrich compound collections with large, goofy, polar compounds, they're probably going to be really good against RNA, but won't necessarily solve the problem against sort of protein bacterial targets. Almost on the home run now, final section about some sort of assay networks. So one of the original visions for ChEMBL, one of the reasons that we focused on not just the biochemical target data from papers, we tried to abstract everything. So 40% of the data in ChEMBL is effectively this biochemical, this B class, which is a compound with a target. 60% is ADME or functional assays. And what we wanted was a way of sort of mapping between a target and a clinical trial. So one of the things that I've got a PhD student working on now, Magda, is building a sort of automated map from ChEMBL of these interrelated assays, organized along a sort of continuum between biochemical assays, cell-based, functional, animal disease models, then human clinical trials. It's pretty easy to build these assay compound co-occurrence networks. It's also pretty easy to make them directed, so they flow from initial screening through to sort of more complex high-value endpoints. And the idea is that we'll have a series of nodes and potentially models at each of these nodes that will provide both, I guess, performance criteria, so how complex or how potent a compound needs to be in a particular assay to proceed to the next stage, and what are the general properties of screening a target and then finding the eventual human clinical trial, which again is just a sort of pretty expensive assay. With this data, you can do some pretty neat things. So this is a bunch of kinase inhibitors, a chemical space plot, log P, molecular weight. If you filter for active compounds in a cell-based assay, this is what you discover. If you then apply the constraint that the compounds need to be active in an in vivo assay, of course, you effectively rediscover the sort of things consistent with good oral absorption and first-pass metabolism. So the sort of rule of 5 as an imprint across compound property space, rediscovered automagically from just organizing the assays in this sort of cascade. So you can, this is an assay graph of approved drugs taken directly from the ChEMBL data. The clusters have got some pretty interesting properties. So this is an oncology property cluster. If you look at this in more detail, you end up with DNA replication and repair as a distinct cluster from signal transduction and hormones. So that the networks and graphs make some sense. You can start to search compound similarity or target similarity across this if you wish. But one of the things we've had most success with is using Word2Vec to cluster the assays in ChEMBL. So it's a really, really simple, somewhat nominal AI sort of tool to come up with semantic similarity of concepts based on effectively the similarity of the underlying vectors or frequencies of those words across the literature. And there's a little bit of smartness in transforming this into a distance metric. And applying this to ChEMBL, we can pull apart, say, chronic versus acute pain models pretty easily in terms of the underlying similarity. And this was really posed to sort of avoid the development and application of these ontologies. I just don't think there's enough time in the universe to develop ontologies that will explain what we want to do with bioactivity screening and so forth. Things like the BAO are great if you index up pre-existing assays with them. I think discovering that from the literature is a sort of Sisyphean task, and that's why we started to develop this Word2Vec approach. So this is now a PCA of these Word2Vec assay descriptions. So about 80,000 assay descriptions into a 200-dimensional space. The size of the cell or the color of the cell just indicates the sort of population. But the relatively magic thing is that when you take those cells and map the compounds that occupy those cells against therapeutic indications, things make quite a lot of sense. So there tends to be some structuring, so hypertensive, so this funny little group down here. Anti-epileptics, anti-inflammatories, and so forth tend to fall in pretty sensible sort of areas. But the nice thing is, potentially now we can take any arbitrary sentence from the scientific literature and project it into this sort of assay space. If it contains the words that are associated with some of the signals in these Word2Vec projections, we can take an arbitrary bioassay from a patent, say, and project or infer some likely therapeutic application. The other nice thing about this projection, this is now back to a sort of graph view, is that we we do a pretty good job of capturing all of the cidal targets in a single, unique, relatively isolated cluster. So drugs roughly fall into two classes, those that just restore homeostasis, health, and those that kill cells. So antibacterials, antimalarials, antivirals, sort of edge case. But killing cells is clearly very different in terms of the targets and the mechanisms at work than restoring homeostasis. And this graph seems to do this. So, a lot of the work, of course, done while I was at the EBI. This is the group on the day I left. A couple of those people came to join me at Benevolent AI, so Mark Davies, Annalie, and Magda came across when I left ChEMBL. Long-term collaborators, ICR, UCL, CNAG on some of the resistance work that I haven't told you about today. Krista at FIM on some of the kinase stuff, and of course the funders here that made all the work possible. So thank you very much.
So you mentioned that you have a great deal of skepticism to community interaction with or community submissions to ChEMBL, but also you mentioned some reports of errors in ChEMBL that were obviously done by people from the community using ChEMBL. So is there some feedback mechanism where you can use this information and integrate it back into ChEMBL to correct these fished out mistakes?
So I guess to be clear about what I meant, so I think there's more effort spent in telling the rest of the world how clever you are, as opposed to feeding back the data to improve the work. And there's there's quite a few groups now that have got cleaned up versions of ChEMBL and talk about them being their magic sauce and stuff. But why don't they give it back to the community? That's the way to build a reputation. That's the way to build better community value. So that's what I was sort of getting at. I think community depositions to ChEMBL are actually going pretty well. They're small. It's often the sort of large pharma companies that have done this. So AZ and GSK have done some submissions. I think some of the stuff that GSK explored was really interesting, actually. So you don't report all the compounds made as part of a program. And for a couple of papers, I guess, they deposited data for a broader set, often the inactives. So having access to these close-in inactives is a good thing to do. They look pretty poor when you report loads of inactive compounds in a careful JMed Chem story. But it's this sort of curation of the data and the feedback is actually a pretty difficult thing to get right. These communities tend to end up dominated by a small number of very active voices, as opposed to a broad input. So if you do find errors in ChEMBL, mail them, and in the next release they're typically fixed. They can't deal with things like the compounds are drawn the wrong way up, or the ionization state of the carboxylic acid is wrong. The fundamental errors, please send them to the guys.
That might be a somewhat awkward question, but when you were looking at the differences between activities of compounds in rats and humans, and you found this variance, did anyone ever think about looking into where does that variance actually come from? Because the source of the data, there's a lot of pressure on people to get results published. So are there kind of sources, labs, which are more trustful than others?
I don't know. I'm sure, in future there's probably going to be some forensic type. I think a great vision for the future of the literature is, the ability for at either times of writing or times of peer review to be able to say, how does this data actually map to the rest of the world? Is it odd? Do I need to look into methodological issues as opposed to the messages they've come out with? But is that going to happen? I doubt it. Maybe with some of these sort of bioarchive and the future ChemArchive, they will, but it'll be a tiny little bit of data and things. In terms of the trustworthiness of the labs, I think this trust isn't the right word. It's just what you're trying to use the data for. This case here, the cell line screening data, there's now actually, so the large-scale cell line screening data for the anti-cancer setting, there's now something like 5 independent replicates of essentially the same experiment. 4 of them correlate really well amongst the other. It's a little bit like Sesame Street. 4 of these things are kind of one thing. One of these things is kind of the same, whatever it is. I can't remember. It's a long time since I watched it. But there's a clear outlier, and the outlier group have looked and said, hey, our readout probably wasn't the best that it could be. Our data processing wasn't the best that it could be. So we're changing our protocol there. So you know, that's a sort of nice example. But I don't think it does any good to, you know, call people out, you know, who've acted in good faith and things. The other trick or thing we use in ChEMBL is if a number is exactly the same from one paper to another, like 4.332, it's likely to just be copied from that previous paper, and often they are the other sort of review articles and things. So I think in ChEMBL these are flagged as duplicates. And again, so if you have this sort of curve, there's actually a lovely little spike where everything is perfectly the same and unbelievable. So the more, you know, like Benford's law, you know, the SAR data should follow that. It'd be a nice little interesting project for an intern or something. Are there always 1s and 2s at the first digit of bioactivity data, as you'd expect, or are things biased in odd ways? So I think there's a lot of really fundamental sort of data science stuff to do. But you can only do it once you pool all the data together and try and have a hash at integrating it.